Okay, and please start.
Okay, sir. Moi start কৰিছো. A very warm good morning to the August gathering present here. Today, on behalf of the Research Scholar Forum, Department of Zoology, and Zoological Society of Assam, I, Queen Hoikia, Research Scholar of Zoology, Guwahati University, welcome you all to this international webinar. Zebs Kluger once quoted, "Suffering is always hard to quantify, especially when the pain is caused by as cruel a disease as Alzheimer. Most illness attack the body. Alzheimer's destroy the mind and in the process annihilates the very self. Brain, the most complex part of the body, consists of more than 100 billion nerves that control our thoughts, memory, speech, and many other vital functions of our body. For some unclear reasons, nerves in certain brain areas degenerate, causing progressive dementia. Alzheimer's disease is one of the common forms of dementia, which is an irreversible nervous disorder in which brain cells connections and the cells themselves die, eventually destroying memory and other vital functions. Taking this as the core of today's webinar, we wholeheartedly welcome one of the most eminent scientists of the world and our pride, Dr. Utpal Das, Associate Scientist, Department of Neurosciences, University of California, San Diego, USA, who will be addressing on neurodegeneration, stem cells, and the future of brain research. So now I request Anbora to carry on the today's session. Over to you, Anne.
So thank you so much, Queen Soikia, for your wonderful welcome address. Okay, thank you. So now I would like to request our Professor Jyotisundar Kodita, Head of Zoology Department of Guwahati University and General Secretary of Zoological Society of Home, to make his inaugural speech. So, sir, I request you please start with your speech. Inaugural speech.
Thank you, and thank you, Queen Soikia, our esteemed President of Geological Society of Assam, Priyabhatala Hakar sir, and all dignitaries and participants and student friends. As Dr. Utpal Das will be talking from America, so good evening to Utpal and good morning to all of us here. So when he consented on that day, I became very glad that yes, our product, a product of BHU, okay, Banaras Hindu University. Initially, he did his graduation from MC College, Borpeta. Therefore, I am saying that our product, signing in the world, California University, San Diego, United States of America, it's a great struggle. He went through MC College, Borpeta, Banaras Hindu University, All India Institute of Medical Sciences. Then he was in NIH, and to our pride that he is in now in one of the top ranking universities working with very critical issues. As Queen has narrated few things about brain research, neurodegeneration, stem cell, very frontline areas. And once our student, as he has been engaged in this type of work, obviously it is a pride from this great nation, India. And he has a very good networking of the similar kind of workers across the world. So therefore, for a long time, we were eagerly waiting to have Dr. Upal Das meet us. But as you know, he is very busy, but he told me that, yes, sir, one day I will give you time. And recently, few days back, when we talked, he said, yes, probably 25th of June, morning to 10 a.m. will be the best time for him. As we were talking informally few minutes back, he was in the lab and he was working. Now he from work directly at home and he will be talking from home. So I wish that this will be a great success and it will be a great connection with the scientists working in top-ranking universities like California University and Guwahati University, Dibrugarh University, and universities in India. And my dear esteemed participants joining from India and abroad, because yesterday I got confirmation from University Dhaka and also Thailand, Myanmar. And people are joining today, I'm sure. So I think all will be enjoying your talk. Thank you so much, Dr. Utpal Das, for giving us time. And we wish you a very, very brilliant academic future. Thank you.
Thank you so much, sir, for your wonderful speech. Now, I would like to ask Sangeeta Das, Research Scholar of Zoology Department of Guwahati University, to introduce the speaker of our day. So please start with your introduction, Sangeeta Das.
Thank you, Anne. Good morning. Good morning, sir. Good morning, Utpal Das, sir. Good morning, one and all present here. It is my honor and great privilege to introduce before you Dr. Utpal Das, who is presently serving as Associate Scientist, Department of Neurosciences, University of California, San Diego. Dr. Das received his bachelor's degree in zoology from MC College, Borpeta. He did his master's in zoology from prestigious Banaras Hindu University, Varanasi. Dr. Utpal Das has earned the PhD degree from Department of Biophysics, All India Institute of Medical Sciences, New Delhi in the year 2008. Sir pursued his postdoctoral research from Section on Neurophysiology and Biophysics, National Institute of Child Health and Human Development, NIH, Bethesda, and also from Department of Neurosciences, University of California, San Diego, in 2009 to 2010 and 2010 to 13, respectively. Dr. Das has worked as a research scientist in ICMR Bioinformatics Center, AIIMS, New Delhi. In the year 2014, Dr. Utpal Das was appointed as Assistant Scientist in Department of Neurosciences, University of California. He was further promoted to the post of Associate Scientist, which he served from 2017 to 2020. Presently, he is working as Associate Scientist in the same university under a very interesting topic related to Alzheimer's disease. He has been bestowed with numerous scholarships and awards, which include Travel Award, NIH Postdoctoral Fellowship, Young Scholar Award, Alzheimer's Association USA, to name a few. He has also served as a member in numerous societies of USA, including American Society of Cell Biology, Society of Biochemistry, and Molecular Biology. He has published numerous research papers in high-impact national and international journals. He has also contributed in publishing 2 book chapters on Alzheimer's disease. He has undertaken several research projects and completed successfully. He has also been invited to various conferences for delivering talks and presenting posters in USA. This short oration is not sufficient to justify his versatility in the field of research, but we are really blessed and grateful to have such an eminent personality among us. Thank you, sir. Thank you for being with us today. Over to you, Anne.
Thank you so much, Sangeeta Das, for such a wonderful introduction. So now I would request our speaker of the day, Dr. Utpal Das, to begin with his presentation. So, sir, please, you can begin with your presentation now.
Okay. Thank you. First of all, Jogan Kolita, sir, and then coordinators Queen, Sangeeta, and for the generous introduction. And before I begin, I would like to thank Research Scholars Forum, Zoology Department, Guwahati University, and Department of Zoology, Guwahati University, and Zoological Society of Assam. So I have been following JSA for last 1.5 years religiously, and I must say you people are doing great job during this lockdown period. I mean, this is amazing. Not many people are doing that. And personally, I can say you one thing. So Jessica sir actually knows that my family personally. So I have a niece, she's doing biotechnology, master's biotechnology. So I asked her to go over those YouTube channels, wherever, something interesting or related to her syllabus. So people are getting benefited. Keep up the good work. And also I'd like to, thank president, working president, and General Secretary of JSA. So let's begin. Let's discuss it. I must say that let's not do a one-way talking. If you guys want to interrupt me anytime, feel free. Okay, let's make it very informal. So let's see. You can see my slides, right? Can you see my slides?
Yes, sir.
Yes,
Yes, sir.
We can see your slides.
Okay, great. Okay, nice. So great. Like Quinn said, I'll be talking a little bit about neurodegeneration, and she gave nice introduction to neurodegeneration with special reference to Alzheimer's disease. And then, actually, I've been working on this disease since my PhD. At AIMS also, I worked with Professor S. Srinivasan. And then also, I worked on neurodegeneration and Alzheimer's. And later on at NIH, and also since I joined UC San Diego, I have been working on this disease continuously. So I'll be talking a little bit on neurodegeneration and Alzheimer's disease, and then some of the tools and techniques we are using to study neurodegeneration, such as stem cell technology. And then a little bit on future of brain research as such in a broad way, where we are standing now and what will be the next step. Okay, so another thing, I don't want to bombard you with a lot of data and thousands of graphs, otherwise people will sleep. So I'm taking it, very lightly. It's a superficial way. If you have any questions, feel free to contact me later on to know more about what I am doing and what's the future of Alzheimer's disease. Okay, so let's begin with the human brain. So, adult human brain, it's around 101.5 kilogram average weight. And if you take a slice of human brain and look at under the microscope, what we see is nerve cells or neurons. And as I mentioned, we have nearly 100 billion, that corresponds to 10,000 crores of neurons inside our brain. And all these neurons, what they do, the main thing is to communicate with each other. Otherwise, they're of no use. They do not, generate any energy to run other organs or our hands or legs or anything. So all they do is communicate with each other. And those communication points are called synapses. And interestingly, we have 100 trillion connections or 100 trillion synapses that correspond to 10 lakhs crore of synapses inside our brain. So just like any other organ, human brain also follows some division of labor, but division of labor in a coordinated manner. It's not just random division of labor. So what happens inside the brain, we have different regions with different colors. I'm showing here, let's say frontal lobe. The frontal lobe, it does the speech and it's essential for smelling and planning and problem solving. Then we have here cerebellum, which is essential for coordination, occipital lobe, that is essential for vision. Then we have big cortex region. And this cortex region, or basically temporal lobe, it also contains a tiny region, it's called hippocampus. And hippocampus is the CPU, kind of. It's essential, or that basically is the storehouse for memory, that is essential for learning. So different regions are there doing their individual job, but all of them in a coordinated manner. But just like any other organ, human brain is also susceptible to different diseases. And when we call diseases, most of the time what we think, somebody has got brain cancer or somebody got epilepsy, right? But besides, of course, those diseases are very common, but besides that, human brain suffers from a couple of other diseases. And these are the list of diseases which we are kind of nowadays focusing on. Let's say multiple sclerosis. What happens in basal ganglia region where these green dots are there, here and here? What happens? Neurons that contains a myelin sheet. That myelin sheet is kind of degraded due to an antibody generated by our own body. So what happens? They become demyelinated and they kind of eventually die, those neurons. And that's how we call that person is getting multiple sclerosis. Similarly, Huntington's disease. So Huntington's disease, what happens? There's a protein called huntingtin. So huntingtin is basically, it extends its length of the protein automatically. It's kind of autonomous disease. So what happens once that person gets this disease that also causes neuronal loss or neuron death? Then it's very common disease. Every now and then, probably you have seen people with checking palsy. So it's called Parkinson's disease. So Parkinson's disease, what happens in this region, substantia nigra and cortex, a small protein called alpha-synuclein that aggregates inside the neuron, and that's how it inhibits dopamine secretion from dopaminergic neuron, and that's when a person actually becomes shaky. That person cannot move. It's a kind of movement disorder. And then finally, we have Alzheimer's disease. All of you, I'm sure you have heard about Alzheimer's disease, which basically, occurs in the hippocampal region here, destroying the hippocampus. But all these diseases I mentioned, people have, or clinicians have kind of clubbed them together, and we call that dementia. So dementia is kind of a general term for loss of memory, language, and problem-solving ability. This is not actually disease. This is kind of a symptoms of a vast range of diseases. So today we are going to discuss a little bit on Alzheimer's disease, and most interestingly, Alzheimer's disease.
Utpal, you please unmute. Utpal, you are muted. I am.
Can you hear me now?
Yeah, now.
Yes, sir, hearing. Yeah, sir.
Now.
So how far I should go back, or
Unmute.
should I continue?
You go back, okay, maybe one step back, then you continue.
Okay, let's see. You can see my slides now?
Yes, sir, we can see your slide.
Yes, sir.
Okay.
Yeah, it's okay from here.
Okay. So all these diseases, clinicians basically what they do, they kind of generalize these diseases, and as a whole we call dementia. If somebody is having dementia, that means that is characteristics of loss of memory, language, and problem-solving abilities. But today we are going to focus largely on Alzheimer's disease, which leads to irreversible memory loss. So what I was saying that during lockdown time, many of you are watching movies, so try to watch these movies. So all these movies, what I'm saying is that these movies were based on Alzheimer's disease. One or more characters in these movies, they had Alzheimer's. So why I'm saying that one should watch these movies is not just to see what happens to an Alzheimer's patient, but also what happens and how the family members suffer if somebody has Alzheimer's in their family or at home. So these are must watch, I would say. So why it is important to study Alzheimer's nowadays? It's because there is a good news. The good news is, if you see, the life expectancy at birth in Indian population, and look at the graph is going up. So in 1960, the average life expectancy in Indian population was just above 40 years. And in 2018, this is just, I took the NITI Aayog data and World Bank data, it went up to almost like 70 years. So this is a good news. People are, we expect more lifespan. Of course, still it's below United States and China, but we are not bothered about them. But what I'm saying that the last kind of 60, 70 years, that life expectancy of Indian population has went up to almost like 75%. And the reason behind this increased lifespan is because many of the dreaded diseases we see within common people, their occurrence has gone down. Look at these diseases like breast cancer. Only there is an increase in last 10 years, only I would say 1% increase. Prostate cancer 1.3. And interestingly, heart disease, strokes, they went down. So basically within last 20 years or so, the percentage of death due to these particular diseases went down significantly. Look at HIV, almost negligible now. So all these are good news. But unfortunately, here comes the bad news. The bad news is, look at the death, the increase in person death due to Alzheimer's disease. Of course, this is worldwide data, went up to nearly 150%. Because life expectancy is increasing, because people are getting cured. Medicines are there for so many diseases, but there is no cure so far for Alzheimer's disease, and people are dying because of Alzheimer's disease or die with Alzheimer's disease. So that's the reason why we are studying Alzheimer's disease worldwide. Now you can say, oh, okay, Utpal, you are in US and you are working for kind of a Western disease. It's nothing to do with Indian population, but that is wrong. Why it's wrong? Because aging is the major risk factor for Alzheimer's disease. And above 65 years of age, the risk of Alzheimer's doubles every 5 years. And if somebody is living beyond 85, then out of 3 people, you will get 1 person above 85 with Alzheimer's most likely, or some kind of other dementia. And in India, we have 130 crores population, and out of 130 crores, nearly 10 crores or above are above 65 years of old, or maybe more than that. And according to 2025 data, India has kind of almost like 50 lakhs of Alzheimer's or dementia patients, which is going up. Almost, it's going to be double in next 8 to 10 years. The problem here is in India, we don't really bother about Alzheimer's. It's still a hidden problem. And many of us, I have seen in my families, that many of us, we just think that, okay, memory loss is nothing, it's not a disease, it's an inevitable part of aging rather than sign of a disease. So these are the reasons why we don't really see Alzheimer's patient, but actually it's there and it is increasing. So that's the reason why we need to study also in India. Now, you can ask that, okay, I have started forgetting things. Am I having Alzheimer's? No, you are not. The thing is that many of the symptoms or signs of Alzheimer's and dementia, they kind of overlap with typical age-related changes. Let's say somebody, many of you probably forgot where did you keep your car key, which is okay, fine, that is not Alzheimer's. But if you are forgetting that consistently, then that's a sign of dementia that could lead to Alzheimer's. Sometimes we forget words, sometimes we forget human name or friend's name. That is okay, totally fine. But if you are having issues with conversation, or you lose track of date or season, misplacing drink consistently, that's a kind of sign of Alzheimer's. So it's very thin line between the signs, but clinicians at least can evaluate. So one should not neglect it. Now, if we talk about Alzheimer's, it's not like that in 1 year, somebody will get all the symptoms. So if clinically, what clinicians, they divide Alzheimer's disease like in a couple of different stages, early, middle, and late stages. All the signs I just mentioned, they start appearing at the age of 60, 65, right? So there is a thin line between mild cognitive impairment and Alzheimer's disease. But eventually when those symptoms increases, then, people will start getting delusions, they get restless, they get agitated, and then little bit of bladder and bowel movement, then issues, they start appearing. That's where, these signs clinicians term at the middle of the disease. And then eventually, those signs you will see visibly. So people actually, they lose complete control of bladder and bowel movement. And, what happens, then people will forget their own name, they will kind of unable to recognize son or daughter or wife or husband. And many times what happens, you can try to feed them with your hand, but they forget how to swallow. So those are secondary effects, not directly related to brain or brain disease, but these are secondary effects start appearing. But interestingly, from early to late, this phase is very fast. Within 5 to 6 years, you will see this kind of changes happening in your near and ones. So it's dangerous situation, and one should take care of early symptoms. But unfortunately, you can ask, Utpal, is there any biomarker? Can I go to a clinic or let's say Guwahati Medical College tomorrow and test for, Alzheimer's, I'm having Alzheimer's or not? But unfortunately, no. There is no available or standard biomarker so far for Alzheimer's disease. Many research labs, many private companies are working on it, but still it is in budding stage. All clinicians, they do neurologists, they will ask you questions, And if you can answer, good. If you cannot answer, they will say, oh, this person is having dementia. Another thing what they do, that okay, they will put you in an MRI tube, or they will do positron emission tomography. And then what they do, they look at the brain volume, if there is a decrease in brain volume or not. But, decrease in actual brain volume, what I just showed in brain atrophy, that takes place at later stage. By the time, it's already you are in the middle of your Alzheimer's. So actually MRI doesn't really help. Similar thing goes with PET scanning. So there is no standard diagnostic tool as of now for Alzheimer's. That is one of the problem why somebody is diagnosed but already at the middle of this, and there is no cure for that. So what about the risk factor? Why some people do get Alzheimer's? As I mentioned, aging is most important and most prominent risk factor. But other than aging, you have other environmental factors such as diabetes. High diabetic people, there is a probability they will get Alzheimer's. Food habit, if you kind of go every day or have breakfast, lunch, dinner at McD or KFC, there is a good probability you will get Alzheimer's because of food and lipid content. Then stress, that is another problem. And little bit of genetics. Only 5%, I would say, or less than 5% of people actually, they inherit Alzheimer's disease. There are mutations in 3 or 4 genes have been identified by GWAS studies. So only 5%, but 95% of people are sporadic. They can get Alzheimer's anytime. And then some of the people with traumatic brain injury, let's say, bomb blast or some accident, there is a possibility they might get Alzheimer's as well. So now, what actually happens to an Alzheimer's brain? So this is a slide I'm showing. This is little gross. Excuse me for that. So you can see here, this is a healthy brain here, right? You have nice corpus callosum here, nicely finger-like structures all over the brain. And this is an Alzheimer's brain here. And you can see the atrophic brain here, this kind of an empty space compared to the normal healthy brain. And if we just take a cross-section of human brain here, you will see this nice compact cortex in healthy, but you see the empty space in the Alzheimer's patient. And this empty space here where hippocampus is located. So now you can imagine there is no hippocampus in a human brain, then what will happen to that person? Obviously, he will forget his own name. So this is what, apparently what we see in an Alzheimer's brain. Now, as the time progresses, this is what we see. Now, thing is, what happens at molecular level? What is the change? What is the bad thing happening inside the brain? So what we can do, if you take a slice of an Alzheimer's patient brain and look at under the microscope, what you will see some kind of deposition, this kind of extraneuronal deposition, extracellular deposition we call. And these depositions are called amyloid plaque, okay? So next thing is, why these amyloid plaques are responsible, what it is doing inside the brain. So this is what amyloid plaque does actually. So in healthy neuron, like I showed before, you have connections, 100 trillion connections, synapses. But these amyloid plaques, which are extracellularly deposited, they kind of kill these synapses. That's why they are called synaptotoxic. So those connections are broken down, and finally neuron will die here. So there is no cell-to-cell, neuron-to-neuron communication, and this communication is disrupted by these amyloid plaques. So now you can ask me, what is the origin of amyloid plaques? Where from all of a sudden these plaques appear inside the brain? So these amyloid plaques, these are made up of a small peptide, small polypeptide. All of you know, right, peptides. Those peptides are called amyloid beta peptide, or in short, A-beta peptide. And interestingly, these amyloid peptides are produced from a parent protein called amyloid precursor protein, or in short, APP, amyloid precursor protein. Every human, including JSA members, all of us, we have APP in our brain. So this APP is a single-pass transmembrane protein. And what happens in normal brain, all the time, right now I'm speaking, this is actually happening inside my brain, or same thing happening in our Jessica sir's brain as well. So what's happening here, APP first is being cleaved by one enzyme called alpha secretase. And because of this alpha secretase cleavage, 2 peptide fragments will appear, generated. APPS alpha and C83. And this C83 peptide fragment is further cleaved by another enzyme called gamma secretase. And it will produce two different peptides, P3 and C59. So this is a good thing. These peptides are good guys. They don't really do any harm to your brain or a neuron or a synapse. But something happens in an Alzheimer's patient or who are predisposed to Alzheimer's patient. What happened, instead of alpha secretase, this APP is cleaved by first one protein enzyme called beta secretase or base 1. And that releases two fragments. Instead of S-APP alpha, it releases S-APP beta and C99. So it's slightly longer peptide. And this C99 is further cleaved by gamma secretase. And due to this second cleavage, it will generate amyloid beta peptide and C59. So this amyloid beta peptide in Alzheimer's brain, that's how it's being generated. That first, instead of alpha secretase, somehow, we don't really know why, it is now processed by beta secretase or base, and that's how A beta is generated. And this A beta, once generated, it forms those neurotoxic or synaptotoxic amyloid plaques. Now, if you look at the sequence of this, so this is what we usually see. It's a very tiny polypeptide. It's just 42 amino acids long. But if you look at the sequence carefully, it's enriched in highly hydrophobic amino acid residues. So due to their hydrophobicity, they form kind of oligomers or aggregates. This is their biophysical characteristics. So there are so many hypotheses floating around what causes Alzheimer's disease, but this is one of the most common and accepted, that this A beta, they form kind of oligomers, and those oligomers, they bind to the synapse, and that's how it inhibits synaptic transmission or synaptic neurotransmitter release, and that's how it inhibits long-term potentiation. And not only that, it also induces reactive oxygen species, it alters different kind of signaling pathways, impairs neuronal protein transport, and also tau hyperphosphorylation. So there are a cascade of things is being done or triggered by this bad guy here, amyloid beta. So why amyloid beta is important, why APP is important, this is another example I am going to give you. You must have heard about Down syndrome, right? So there is a great connection between Down syndrome and Alzheimer's disease. So you have seen this cute little kids. Many of you, I'm sure you have seen that they have Down syndrome. What happens in Down syndrome, there is an extra copy of chromosome 21. And interestingly, the APP gene, what I was talking about in previous slide, is located in chromosome 21. So what happens in Down syndrome, there is an extra copy of APP gene. That means there is an increased APP protein and increased A beta level in Down syndrome. And amyloid plaques are observed in Down syndrome patient at the early age actually, at like 30s, late 30s or so. And almost like 50% of people with Down syndrome, eventually they develop Alzheimer's disease. I put a stars in here because average median lifespan of Down syndrome people is around 55 years. And that after that, they usually don't survive. But at that age, if they survive up to 55 years, then 50% of them will get Alzheimer's. But luckily, if they survive for another 10 years, let's say 65 years, then almost 95% of those people will get Alzheimer's. And in India, this is the statistics I just generated from DS Foundation of India according to 2018 data, that almost like 1 in 900 live birth is having Down syndrome. Almost like 30,000 children every year they born in Down syndrome. So imagine almost 50 or 60% of children eventually will get Alzheimer's in India. So things are changing. We need more importance to this kind of diseases. Now, let's go a little bit more deeper into the production of amyloid beta in neuron. As I said, something happens to Alzheimer's patient brain and APP is then cleaved by beta-secreted enzyme, right? Now, if you just remember our class 8 or 9 biochemistry or biology book, this is what we see, right? For any enzymatic reaction, there is a substrate and an enzyme, and there is an active site. So substrate comes and binds with enzyme, and then two products are produced, and enzyme is released. Just imagine same thing is happening inside our brain all the time, including JSA members, right? That APP is there, which is a substrate. Base is dienzyme. APP and base interact, and SAPP and beta CTF or C99 is being produced, which is finally that this guy beta CTF produces beta. So this is kind of a rate limiting step of this entire Alzheimer's disease progression or generation. So as this is a rate limiting step, various pharmaceutical companies, many research labs, they are focusing to understand better this particular interaction or the association of APP and the base. So we are not different. So what we wanted to know, first of all, inside the neuron, where these two proteins live inside the neuron. And then what happens that all of a sudden base and APP start interacting and kind of the amyloid beta production starts. So as I said, we wanted to know in a neuron. Why? Because in the last 5 or 30 years, many people have published hundreds of papers describing, APP and base localization inside the cell. And those started, when I'm talking about site means I'm talking about subcellular organelles like APP or base, whether they reside in endoplasmic reticulum, they're in Golgi, they're in plasma, they're in plasma membrane or endosome. And if you see the list, it kind of covers all the different available organelles inside the cell. But interestingly, if you see their model, what they're using, most of them were not even neurons, some neuronal cell line or different other cells. Very few of them actually use neurons. So we wanted to test these things in neuron because non-neuronal cells, if you see apparently, they're pretty different from a neuron. So this is a non-neuronal cell. It's a tiny little cell, triangular, you have a big nucleus here. But if you look at the neuron, see its morphology first. It has a nice big cell body, which resembles to a non-neuronal cells, but then it has hundreds and thousands of primary and secondary dendrites emanating from the cell body. And then it has a long, beautiful axon, which can be sometimes, 1 meter long. So whatever the results we see in non-neuronal cells, you actually cannot just say that something is happening inside a neuron because metabolism, their organizational structures, they're pretty different in neuron. So we wanted to test where APP and base lives inside the neuron. So obviously, first thing you can say, where do you get neurons? So this is where we get primary neurons. We take mouse. I'm sure many of you are using the same tools. So what we do, you take out the mouse from either uterus or the dead bone, and then cut open the brain and just direct out the hippocampus. And those hippocampus we just culture. And this is what actually you look at under the microscope. Dead means days in vitro. This is what neuron look like when they just took out from the brain. And after 1 day, 2 days, 3 days, you will see after 14 days, these nice processes are kind of emanating from the cell body. And if you stain with certain markers like MAP2 and tau, you will see beautiful neurons. So, okay, we are ready. We are doing these things every day in our lab. We have neurons. Now, next question you can ask, Utpal, how are we going to look at the proteins you wanted to look? So our main target proteins are APP, amyloid precursor protein, and beta secretase, base one. So how to visualize this protein inside the neuron? So we needed to develop some tools. The tools many of you are using probably is that use molecular cloning and recombinant DNA technology, where, what we did actually, we took APP gene, we took base gene, and fused to a couple of fluorophore. In APP, we kind of fused green fluorescent protein, and in base, we tagged M cherry or red fluorescent protein. And what happens when you have this DNA, these are now recombinant DNA, you transfect them with certain reagent and wait for a couple of hours, and then you will start, cell will basically transcribe and translate these two different proteins, but now these proteins will be colorful. One with green color, another will be red in color. So this is a dendrite, segment of a dendrite I'm going to show you. So on this side, left, you will have cell body, and this side is a dendritic tip. And all this green, all these white dots inside this, they're actually green. Okay, they are green. But look at the movement, how they move inside the neuron. So see this video here. So this puncta, these are APP, GFP, bidirectionally they are moving. One of them are moving this way, that way. Look at this, how they're moving inside the neuron. They're beautiful to see actually. So We can visualize, we have neurons, we have tools to visualize. Now, next obvious question was to, look at the actual organelle. And for that, what we did, we expressed both APP, GFP, and base M cherry simultaneously inside the neuron and did dual spectral live imaging. And this is what we see. Actually, remember, this is the same dendritic segment, but I have separated spectrally so that you can visualize two different proteins easily. The green dots are APP, red dots are base. See what happens. So both of them, they are transmembrane protein. Remember, they are being synthesized in endoplasmic reticulum, and then they move in different organelles probably. So they are moving bidirectionally from cell body to the dendritic tip, and they're coming back. So this is what they do. That's what they're meant for. And how to analyze this data? We are seeing some movie, but how to interpret the data? So to do that, what we do, we draw chymographs. So chymographs, basically in these chymographs, what we are showing that these different lines are there. You can see from top left to bottom right. These are kind of lines are because generated from these moving guys. So from top left to bottom right, they are anterogradely moving. That means from cell body to dendritic tip. And from top right to bottom left, they are retrogradely moving. And some of the lines are stationary here. They don't really move at all. So this chymograph, this left part is for APP and right part is for base 1. And then when we overlaid them, what we found that only 20 to 30% of APP and base are together. Remember, they are enzyme and substrate, right? They are actually not together all the time. So obvious question is, what is this transport carrier? Who is carrying APP? Who is carrying base inside the primary hippocampal neuron? So to do that, what we used, something very similar, but this time we use different standard, heavily used organelle markers. Let's say GALT, and these are kind of trans-Golgi marker, and then RAF5, which marks early endosome, LAV11, recycling endosome, transferrin receptor, recycling endosome. So different markers we have used to look at where APP and base moves. And this is what we have observed. When we co-express base with recycling endosome marker transferrin receptor, we saw that almost like 70 to 80% of base are actually moving in recycling endosome, but not with early endosome. So base is at a resting state, base is localized in recycling endosome. So all of them are kind of overlapped. What about APP? What we noticed that APP actually resides in Golgi-derived vesicle. This NPY, this is basically red dot here, green dot stands for APP, and red, this line stands for Golgi-derived marker. So most of them, almost like 80 to 90% of APP are actually inside the Golgi-derived organelle. So this is what, in summary, we see. Base is in recycling endosome, APP in Golgi-derived organelle. And at a resting state, APP and base interaction, that means enzyme and substrate interaction, it's a low frequency event, right? It's not many of them are together. So probably this is why not everybody is getting Alzheimer's. That bad guy base is not associated with APP. Otherwise, all the time you would expect amyloid beta production, and all of us, we would have got Alzheimer's by now, right? So nature kind of has developed a way to keep them segregated. So that's why not everyone is getting Alzheimer's. But you can say that, some people are getting Alzheimer's. They do get Alzheimer's. Why? What happens all of a sudden? And that's where, we asked our second question. What happens inside a neuron that initiates APP-base interaction, which are largely segregated? And we did a lot of literature search to just find out if anything known in, as a trigger for Alzheimer's disease in normal human being. And we come across many things. And what we found that in some brain area are highly activated even when one person is kind of resting, not doing anything focused. So those brain regions or hyperactive regions are called default mode network. So basically this cortex and some of the cerebellar regions, they in blue color, these are kind of default mode network. So they are activated because that's why we are seeing blue color. They are activated when you are sleeping or you are not doing anything significant. So In another way, we can say that if you're kind of a daydreaming or becoming rich or something like that, those regions become activated. So Randy Wagner in 2005, what they showed that those regions which are under default mode network, those regions are observed with high concentration of amyloid deposition. That means whenever there is an increased activity in default mode network, that person could have generated Alzheimer's, or there could be increased A beta deposition. This is what I'm saying. Now you can say, oh, neuronal activity is good. Then how can you say neuronal activity is bad? Actually, you're right. Neuronal activity is good if you are doing some task-specific region. That means, okay, you are studying or you are doing an experiment, you are so focused. That is kind of a very focused way of activating certain regions, this blue color here. But if you're kind of a daydreaming all the time, doing random different things, then those regions which are activated, that is kind of a bad thing. So what we hypothesize that, okay, if you induce neuronal activity, probably that can trigger APP and base interaction. And that's what we wanted to test. First of all, can we induce neuronal activity in cultured neuron? And second, what will be the consequence of this kind of APP-based interaction? So how do we induce neuronal activity in cultured dish? And that's what we did. Actually, there are very standard published protocol. So what we did on top image here, you can see neuron dendritic region transfected with APP green and base red. And this is the overlay. Red and green, they are mostly separated. Very few yellow dots indicating they are interacting. But when we treated those neurons with glycine is a neurotransmitter, right? Just for 5 minutes, and then we observed, look at the amount of yellow dots here, indicating there is a significant increase in APP and base interaction in those neurons when we stimulated them with glycine. And not only increased colocalization or increased interaction, but also there is an increase in beta CTF level, which is a product of base cleavage of APP. That means, we can induce neuronal activity in a dish, and we can see an increased APP-based interaction. Now you can see, you can tell that Utpal, APP, you are saying that they are residing in a Golgi-derived organelle, base is a recycling endosome. How come they come together when you induce neuronal activity? So I would say there is two possibilities. One, that homotypic fusion, two different organelles can fuse together, and that's how they will come together, but which is not possible. We have tested neurons, nothing happens. Another way is APP goes to the plasma membrane and then endocytosed. Endocytosed where? In a base-containing recycling endosome. And how did we prove that? If you inhibit endocytosis with clathrin-mediated endocytotic inhibitor dinosaur, we can basically distinctly inhibit this endocytosis. And this is the result. In a control condition, APP and base, red and green, are separated. When you induce neuronal activity, we can see increased yellow dots representing increased interaction. But when we inhibited endocytosis prior to neuronal activation, we could not able to see any increased interaction. So that means endocytosis of APP from Golgi to recycling endosome is an essential step in generating APP-based interaction or A beta production. And we also use some genetic markers to inhibit APP interaction. So we published this data in last couple of years. This kind of good thing for us. And we kind of first time we said that this is actually molecular mechanism, what's happening inside the cell. That's how, probably these steps are being studied. But unfortunately, you can say that, Utpal, you did all your experiments in mouse neuron, and probably you are right. And that's why so far there is no therapies, because all the data people are publishing or producing or pharma company are doing, most of the time they will use either non-neuronal cells, or people like us who will be using mouse neuron. But if you see really carefully, we are not big mouse. Humans are human, right? If you club hundreds or thousands of mouse, we cannot that will not be equivalent to a human. And Alzheimer's disease is uniquely human disease. We have nearly 10 different transgenic Alzheimer's model in our lab, but none of them actually replicate actual Alzheimer's disease, because mouse are not born to get Alzheimer's, right? On the human. So we kind of need an alternative to a mouse model. So how do we get human neuron? Can we get human neuron and do our studies? And that's probably the ultimate way of doing. But unfortunately, we cannot really do it. So obviously, how main question comes when you come to actual disease mechanism, studying actual disease mechanism, we need human neuron. So obviously, you cannot just cut open a human brain and get some biopsy sample and do your cell culture work. We can open a liver or a lung or a kidney, but brains are not allowed. Don't try this at home. So even when you can say that you can do a biopsy of a human brain and then culture those neurons, but it will be extremely difficult. So next obvious option left for us is, can we actually make human neuron in our lab? And actually we can. So making of a functional human neuron. So for that, what we are doing now, we are taking skin cells or fibroblasts from adult human, anybody, it can be you, me, whoever. And then we kind of take those skin cells and then transfect them with 4 different transcription factors, OX, SOX, KLF, and MEK, and then wait for 3 weeks time. Just wait for 3 weeks. And after 3 weeks, you come to your lab and you will see different kind of cells now on a dish. It's called pluripotent stem cells. And as we are inducing, it's called induced pluripotent stem cells. And what will happen once you have pluripotent stem cells or iPSCs, you can add a couple of inhibitors for certain signaling pathway. That will take another 15 days and you will get neuronal stem cells or NSCs or NPCs. And to those NPCs or neuronal stem cells, if you add certain factors like BDNF and GDNF, after 3 weeks time, you will get a human neuron in your lab. So this is very easy, very easy. Everybody is doing in our lab. But for these easy steps, Shiny Yamanaka and John Gordon in UK and Japan, they got Nobel Prize in Physiology and Medicine in 2012. So they optimized all these different procedures. They found out what transcription factors are essential. Right now, we are just doing, every other lab is doing this process, starting from skin cells to a normal human functional neuron. So once you have a neuron, let's talk a little bit more about this iPSC technologies. And all of you, I'm sure, at graduation or even before that, you have read about human fertilization and development, right? So we have a oocyte, which is fertilized by a sperm that forms generates a morula stage, and that stage later on becomes a blastocyst. So this blastocyst, they call few hundreds of cells, they are called innermost cells. And those innermost cells, they're pluripotent. They can differentiate into 200 different types of human cells. And those cells actually makes heart, lung, neuron, whatever you want. So basically, these pluripotent innermost cells, they have the capacity. So they are called embryonic stem cells. So these cells are also the source of human neurons. So many people in like 2000, early 2000, they started, using this human embryonic stem cells collecting from human fetus. So obviously, as expected, that has even the therapeutic potential, but it has a lot of ethical issues. So, funding kind of stopped. You cannot use human embryos because human embryos are human, right? Eventually. So government stopped, funding those kind of research. Then people started, Yamanaka and all, they started looking for alternatives. So can we, instead of inner cell mass from a fetus, can we generate, or can we make pluripotent stem cells from somatic cells like skin or blood cells? And that's how this cell reprogramming technology has evolved. And nowadays, it's kind of so common that every lab is now reprogramming human somatic cells and making iPSC neurons. So I'm going to show you one video. I don't know if you can run it or not. So this video, you can see here how neurons are made in the lab. Look at this. So this is a neuron. All of a sudden, you kind of generated. That's how they're being generated. So I can run it again. So they are being generated from iPSCs. All these are iPSCs. And once you add different factors, you will kind of have this particular neuron here. So this is how neurons are being made, human neurons are being made in our labs. So within the last 5 years or so, this field has kind of rapidly evolving. So not just human neuron from skin cells, we can generate different kinds of human neuron. Let's say cortical neuron, dopaminergic neuron, motor neuron, different kind of neurons, whatever regions you want, you can make. And not only that, nowadays we are kind of directly taking skin cells, and instead of making iPSCs, we are directly kind of converting skin cells into neurons and astrocytes, microglia, oligodendrocytes. So whatever cells you want, we can make within like 3 weeks time if you give us some skin. Okay, so now this technology has developed, and I wanted to go back to again the same questions, that what are the regulators of APP endocytosis? Because previously I showed you that APP endocytosis is essential for A beta production. So I did that in mouse, but now I wanted to do it in human neuron using the same technology. And luckily, Larry Goldstein, he is a pioneer in our institute, our UCSD, who kind of first used human iPSC neurons from Alzheimer's patient. So what I am doing right now in Larry's lab, I am taking skin cell from normal human, and I am also taking skin cells from Alzheimer's patient, actual Alzheimer's patient. And then I am using all those stem cell technology. I am kind of producing human neuron for my studies, and I am studying these different, endocytic inhibitors. And we have published using the same neurons, what all the different regulators, how they are being modulated, how cholesterol-reducing drugs can rescue Alzheimer's effect. I can discuss all these papers later on whenever you want. And also screening for small molecule inhibitors or future therapeutics using the same technology. I'm not going into details. But here I would like to take another 5 minutes just to show that what all the future of this stem cell biology or iPSC-derived neuron can be used. Nowadays, not just in Alzheimer's, many patients are being injected with iPSC-derived neurons. Some of them are like multiple sclerosis or ALS or spinal injury, and they are getting cured. Clinical trials are still going on. And in Japan, last year, Parkinson's disease patients are being injected in the brain with iPSC-derived dopaminergic neuron, and people are getting cured. But obviously, this is kind of a not very well-defined things. We still need a lot of work because we need to cross this valley of death. We need good scientific rationale. We need better preclinical study, and then only we can go to clinical trials. We are using human neuron, but there could be some side effect as well. So we need a better model. Instead of a two-dimensional neuronal model in a dish, nowadays what we are trying to create, and other people have already generated, that if we can, generate a brain in a dish, not just a couple of neurons, but a brain in a dish. And to some extent, many people have successfully made brain organoids. So these round structures you can see, they are kind of a pea-sized shaped human neuron clusters derived from iPSC technologies, and they are called brain organoids. So these brain organoids within like 80 days of time from one single iPSC, we can these mini brains nicely generated in our lab. And they have better survivability, and they are giving us better opportunity to study synapse formation or a better disease model. But again, this is not the end. We still need to, give, these organoids with blood and other supplies, which are obviously essential in human development. You can see that in a fetus, within like 10 weeks after gestation, blood circulation starts. So similar thing we wanted to replicate in human organoid, brain organoid. Can we kind of supply blood, to these organoids? And actually, we are doing that nowadays. We are basically making human organoid with blood cells, and those blood cells are kind of generating small capillaries and supplying nutrients to those organoids. So we have an obviously better model than what we had, right? So all these technologies evolving, we are kind of now transplanting these, human organoids into mouse, and we are watching it whether mouse can, make friendship with this human organoid, and kind of they incorporate this brain structure with their own brain. And to some extent, people have successfully implanted brain organoid in the mouse brain. So obviously, things are evolving, rapidly evolving. Now, one last question you can ask me, you have a brain organoid. Can those organoids, can you give consciousness to those brain organoids? On any other word, can this brain organoid feel pain or emotions? And very interestingly, very recently, Alison Mottry in our university, our next door neighbor, they published a paper in 2019 showing that these cortical organoids, brain organoids, they kind of express similar gene pattern like what we see in human fetus or neonatal brain. They did electrophysiology experiment and then they looked at EEG or electroencephalogram, and they found that those brain organoids in a dish, they behave like something like neonatal brain. So it's very interesting, a lot of things going on. I mean, we just need to wait for a couple of years. Now, one last slide I would like to show you that we're kind of crossing the barrier in neuroscience. That, okay, we have organoids, but can we replicate a neurological disease-like condition in an organoid? Unfortunately, these neurological diseases like Alzheimer's and Parkinson's, they take years. Like I said at the very beginning, you will see symptoms in 60 years. So we need to kind of expedite the disease process so that we can see them and we can mitigate them. So what people are doing, like Alison Mottry, what he's trying to do, that these systems cannot be replicated on Earth. So he wanted to do that where, this can be sped up, and microgravity is something where this like this kind of conditions can be, propagated very fast. So in astronauts, what has been shown that artery wall becomes stiffer and thicker, like what we see in older people on Earth. So basically what he thought that if we can make some organoid in space, we can better model a human disease, especially neurological diseases like Alzheimer's or so. So what he is doing now, he's collaborated with NASA, and what he did, he sent some of his organoid to International Space Station. And obviously it needs a lot of hardware and software, but what he is doing there, that he sent 1 month old brain organoids to space station, kept them for 27 days, brought back to Earth. And now what he is doing gene analysis. And what he found that although morphology doesn't change, there are some changes taking place, like what he found kind of compatible or comparable to early neurological disorders. So I'll stop here, and this is the summary. I'd like to refocus again that Alzheimer's disease, remember, this is a pathology. This is not normal forgetfulness. So if you see something, you should take care. You should take your keen and near and dear ones to a neurologist to see what it is. And then at a molecular level, what we see at a resting state in human brain, this APP and base, substrate and enzymes, they are segregated, and probably